Hello, everyone. My name is Yihao. I'm a second-year PhD student from Auger Research Group. I'm a second-year PhD student from Auger Research Group, Department of Chemistry, University of British Columbia. Here, I'm presenting our recent published project, a semiconducting polymer directly stabilized with serum albumin preparation, characterization, and cellular immunolabeling. First, what are polymer dots? Semiconducting polymer dots is a nanoparticle made of a long chain of conjugated polymers. They are very brightly fluorescent. And there are several choices have been commercialized for people to use making P-dot and to do a variety of applications, such as bioimaging and medicine and clinical studies. The P-dot chain in P-dot is only held together by weak hydrophobic force. It's not strong metallic, ionic, covalent bonding in variety of inorganic nanoparticles. So they require additional amplifier to give it their extra stability. So the amplifier usually are lipids and epiphilic polymers. However, even though we have the amplifier stabilized P-dot, there are several issues might still happen when we apply P-dot into our studies. The first problem is non-specific binding. Due to the strong interaction because hydrophobic force, P-dot is readily absorbed on any interface, glass surface, membrane surface, and paper surface, even for biological sample surface. Second one is that poor stability, have a narrow range stable in pH and ionic strengths. Once P-dot beyond this range, it will lose their colloidal stability. It's not ideal for the further studies. Third, it has less developed conjugation chemistry. The type of a functional group on P-dot surface is limited. We want to discover more functional group to realize more bioconjugation to connect protein, antibody, and dyes for bioimaging application. Bromine serum albumin, BSA. BSA is a common blocking agent and it can bind many nanoparticles, which means even though we have amplifier coated nanoparticle, when we apply to in vivo or in virtual diagnostic and imaging studies, the protein like BSA in the biological sample will bind onto our nanoparticle. And several studies suggest the albumin-rich corona will enhance the nanoparticle circulation time and stabilities. So our idea is to cut the amplifier. We directly use BSA to stabilize P-dot. We choose FABT, it's one of commercialized semiconducting polymer, and use BSA as coating material. Given several cross-linking process using DSG and JA, we make a BSA P-dot or BSA cross-linking P-dot. The method we make in BSA P-dot is called nanoprecipitation. We dissolve FABT in THF and BSA in water. We use a syringe quickly inject FABT in water solution and while sonicating. After that, we heated up the solution and using nitrogen stripping, evaporate the THF part. So what you're left is BSA stabilized P-dot. And we give the BSA P-dot several washes, remove the excess BSA in water, and go to the cross-linking process. The cross-linker, first one we picked is DSG, disuccinamide glutarate. The mechanism is quite clear. It can interact a main group on the lysine residual in BSA structure and do the cross-linking. And however, glutarate GA, the mechanism is much complex than DSG because they have a multiple formation in the aqueous environment due to different pH and condition. It can make to monomeric cyclic hemiacetal. Also, it can do the auto condensation and polymeric by the others, make to the polymeric alpha beta unsaturated aldehydes. Each different kind of structure give a different result. It's hard to predict which mechanism actually perform in this research. And then we use transmission electron microscope, TEM, and nanoparticle tracking analysis, NTA, to characterize the size we made, BSA P-dot and BSA cross-linking P-dot. They are around 80 nanometers and polydispersed, but the data suggests after do the cross-linking process, the size will be less polydispersed. It's more ideal for the further studies. Now we want to do the performance comparison. We want to compare the traditional amplifier coating P-dot, which is PSMA and PSP P-dot, and a silicon coating P-dot with our BSA and a BSA cross-linking P-dot. Let's see which side have a better advantages. First comparison is fluorescent characterization. We measure the absorption and emission spectra for different type of PDOTs and fluorescent lifetime. And we found BSA and BSA cross-linking PDOT have a similar or more favorable in fluorescent property, which such as have a longer lifetime than the traditional ones. Second, we conduct colloidal stability using aqueous gel electrophoresis. And we want to study the stability of different type of PDOT when they exposed to the wide range of pH from 3 to 8.5. If the certain pH will affect the colloidal stability of PDOT, they will aggregate at the very beginning of the electrophoresis, just like PSMA like that. If the certain pH will not affect too much for PDOT, the PDOT will have a mobility moving downwards, just like BSEG we made. And many electrophoresis data suggest PSMA PDOT have a different level of aggregation when they exposed to the wide range of pH and ionic strengths. However, BSE and BSE cross-linking PDOT can maintain good colloidal stability in ionic strengths and pH for multiple days. Furthermore, for the physical stability, we're using atomic force microscopy, AFM, with fast force mapping, FFM. If the nanoparticle is rigid, it can maintain their close to 1-to-1 ratio of dimension and height when they in the AFM measurement, just like a BSE and a silicon PDOT. And however, PSM PDOT almost flattened because it's soft. It can, after dry on the matrix of the AFM measurement, they will become flattened and a pancake shape. We suggest it's a lossier physical stability under the measurement. And we also did the hardness measurement using FM, and we found cross-linking, it can help to enhance the physical stability for the nanoparticle made. Since we mentioned the traditional amplifier coating PDOT can cause non-subspinning on the variety of interfaces, so we're going to evaluate for that. The first material we pick is polystyrene plate, and we incubate different type of PDOT. It's placed 30 minutes after several washes, measurement fluorescent residual using fluorimeter. And finds compared to PSMA one, the BSA and BS cross-linking one has much less reduced for the non-subspinding. Also, we run the PDOT solution through the nitrocellulose membrane. We want to look at the residual. And we're starting the interesting area, and compared to PSMA one, the BSA and BS cross-linking one have a less residual in the 13 area. So it's, the results suggest the BSA and BS cross-linking PDOT have a reduced non-subspinding on this surface, and is favorable for immunoassay and electrofluor assay for future studies. Also, we study the non-subspinding on fixed A549 cell surface. Clearly, PSMA has an obvious aggregation on the extracellular matrix. However, BSG, we barely see the non-specific binding. Also, for the live A549 cell, silicon and PSMA PDOT have a massive non-subspinding on cell surface. BSE PDOT have a slightly reduced. After due to several cross-linking, we barely see for the BSEG PDOT, and has reduced non-subspinding. The study suggests it's favorable for cellular labeling studies. Before the cell labeling, we do the cell availability using MTS kits, and we incubate the different type of PDOT with alive A549 cells 24 hours. We find a BSE PDOT and have no obvious toxicity to the cell. And we further do the cell uptake measurement. We stain the nucleus by DEPI and lysosome by lysotracker blue dye. And we let the cell uptake the different type of PDOT. We pick PSMA PDOT and a BSAG PDOT. We can see for the PSMA PDOT have obvious patterns similar to the lysosome colocalization. We suggest they can be uptake by cell due to the endocytosis. However, BSEG PDOT, due to they have less non-subspinding on cell surface, they are less prone to be a cellular uptake, is permitting for the cell labeling. For cell labeling, the first conjugation we tried is using ligand receptor binding. We make the BSE and BS cross-linking PDOT conjugated with biotin NH ester to make a BSE PDOT biotin conjugates and incubate with SKBR3 cells. We can say cell is successfully labeled by the conjugates. However, we pre-block the SKBR3 cell with excess biotin and incubate with conjugates with the cell. We see there no labeling. Also, we pre-block the BSE PDOT biotin conjugates with excess avidin and incubate with the cell. We still see no labeling. So 3 experiments suggest the cell might express everything like biotin receptor on cell surface that can directly labeled by BSE or biotin conjugates. The second, the bioconjugation, we try to make the HER2 antibody with tetrazine in NHS ester, and conjugate BSE and BSE cross-linked with transsuccular octane in NHS ester. We want to use click chemistry to realize selective immunolabeling for SKBR3 cells. First, we incubate the BSE JE with the cell. We see there are no non-specific binding. After touch TCO, there are still no non-specific binding. And make a click chemistry happen and incubate with our cell. The conjugates will successfully target the cell, and the labeling is quite efficient. And at this positive control, we use A549 cells. There's no antigen for HER2 antibody, and it makes the click chemistry happen, and conjugates not target on the cell. So this experiment suggests the click chemistry driven selective immunolabeling succeed. As conclusion, BSE is able to directly stabilize P dot, and many improved properties. It has enhanced physical and colloidal stability, and more favorable in fluorescent properties, and has reduced non-cis binding on the variety of interfaces. And it can involve a wide variety of bioconjugates chemistry happen besides tetrazine and transsuccular octane. At the future work, we will apply the BSE and BSE cross-linked PDOT into lateral flow assay, immunoassay, and a smartphone-based bioimaging instrument that's built by my colleagues. I would like to thank all the institutes that on this research and people and facility who helped with this project, and my supervisor, Ross Agar, and all the members from Agar Research Group. Thanks for listening. I'm happy to answer any questions.
Any questions for Yihao? We have maybe 3 minutes left. Okay, Mifi.
Yes. Yes, how many sites that can conjugate with antibody is really depend on the functional group on the BSA. We suggested the amino group can realize NH ester conjugate chemistry. There are around 170 BSE around our PDOT is possibly the maximum we expect over 170 antibodies that conjugate on our PDOT surface. But maybe one antibody is enough for the one PDOT to conjugate to labeling our cell.